Bismillahirrahmanirrahim. Today we are going to learn about the mechanism of action of cortisol. To know about the mechanism of action of cortisol, we are going to focus on these diagrams. First of all, we should imagine that this is a cell on which the cortisol is going to act, and this is a cortisol. First of all, how the cortisol will reach over here in the proximity of the cell. How will it reach over here? First of all, the cortisol will be secreted by the zona fasciculata of the adrenal cortex. Then it will move towards the bloodstream. In the bloodstream, this cortisol will combine with the corticosteroid binding globulin, a special type of protein that is called corticosteroid binding globulin. That is called corticosteroid binding globulin. Corticosteroid binding globulin is responsible for combination with the cortisol, and it transfers the cortisol to the site of action. So this corticosteroid binding globulin will move it towards the capillaries, where the capillaries are adjacent with the are in proximity with the affected cells. So when the corticosteroid reach there in the capillaries, it will detach from the corticosteroid binding globulin and it will move toward the affected cell. It will, as this is highly lipid soluble, it can move anywhere in the body, so it will reach over here toward the cell and it will enter the cell. This is cortisol, and this is the affected cell. This cortisol will reach over here in the affected cell and it will enter the cell. In this diagram we can see that this cortisol has get entered into the cell, into the affected cell. Now what will happen? Inside the cell there is a special type of protein for binding with this cortisol, for binding with the steroid, for any steroid, but especially we are talking about the cortisol. So the cortisol will bind a special type of protein. This is a special type of protein with which the cortisol will combine, and this protein is called a glucocorticoid receptor. This is called glucocorticoid receptor. We can also call it simply as GR, glucocorticoid receptor. It will combine with the glucocorticoid receptor. After combination with the glucocorticoid receptor, what will happen? This glucocorticoid receptor will undergo some conformational changes, and a small protein will be detached from this glucocorticoid receptor. There is a small protein that is called HSP. This is a small protein with the glucocorticoid receptor. This small protein is called HSP. This is called what? This is called HSP. The SP should be small in the subscript. HSP are actually for heat shock protein. So these heat shock protein will be detached from this glucocorticoid receptor due to conformational changes by the binding of the drug. This HSP will be detached. In this diagram we can see that HSP has been detached. When the HSP or heat shock protein get detached from this glucocorticoid receptor, this glucocorticoid receptor is now in active form. This is now in active form. This glucocorticoid receptor will further do in the next diagram. This will combine with another glucocorticoid receptor complex. Now this is called glucocorticoid receptor complex because this glucocorticoid receptor is an active form and it has been combined with the cortisol. So this is called glucocorticoid receptor complex. Now this glucocorticoid receptor complex will combine with another glucocorticoid receptor complex. This is one glucocorticoid receptor complex. This is another glucocorticoid receptor complex. With this glucocorticoid receptor complex, this consists of cortisol and glucocorticoid receptor. This also consists of glucocorticoid receptor and a cortisol. So two cortisol and two glucocorticoid receptor combined together, they form a dimer. Probably by the sulfide linkage, they combine together and they form a dimer. This dimer will now go further. This dimer will now carry on the activity further. How will it carry on the activity? This in this diagram we can see that dimer has been transferred into the nucleus. That dimer has been transferred into the nucleus. When it goes into the nucleus, this dimer of glucocorticoid receptor complex, glucocorticoid receptor complex, dimer of glucocorticoid receptor complex will go to the nucleus. After getting entered into the nucleus, what will happen? What will it do? It will combine with a fragment of DNA. It will combine with a fragment of DNA. In this diagram, we can see that this is the nuclear membrane. This is the nucleus, and in the nucleus this is a DNA strand. In the DNA strand, there is a special type of sequence, or there is a you can say that there is a small fragment of DNA that is called glucocorticoid response element, GRE. That is called glucocorticoid response element. You can simply call it as GRE. You can simply call it as GRE, glucocorticoid response element. You can also call it glucocorticoid receptor element. But simply this is glucocorticoid response element. With this glucocorticoid response element is actually a small fragment of DNA which is responsible for the binding with the dimer, so this glucocorticoid response element will be combined with the dimer of glucocorticoid receptor complex. So after binding with the glucocorticoid receptor complex, what will happen? Now this is very important. In this diagram, we can see that after binding with this is the dimer of glucocorticoid receptor complex, and this is the glucocorticoid response element. The combination of glucocorticoid receptor complex and glucocorticoid response element, these activate the glucocorticoid receptor element or glucocorticoid response element. Glucocorticoid receptor response element is activated, and glucocorticoid response element actually do what? It will inhibit the transcription factor. It will combine with the transcription factor. It will combine with the transcription factor all around. Over here, this is a pathological. This cell is having attack of some pathogen. So this cell is pathological cell. In this cell, some pathological process is being carried out. So in this cell, the DNA sequences are active in the formation of some proteins. This DNA sequence is active. So a lot of DNA sequences are active in the transcription of various proteins which are pro-inflammatory. So what will happen? This glucocorticoid response element will combine with the transcription factor. Those transcription factor which are pathological, those transcription factor which are responsible for the formation of inflammatory substances. For example, I am going to give you an important example of this one. For example, transcription factor are a large number of transcription factors. Some are physiological, some are pathological, but over here the transcription factor should be transcription factor. The pathological transcription factor should be, this should be the best example, KB. NF-KB, this is one of the transcription factor which is responsible for pathological processes inside the cell. NF is for necrosis factor. For example, this is NF-KB. For example, this one is NF-KB. This is a transcription factor. When this transcription factor enters the cell, and it enters the nucleus and it combines with the DNA. After combining with the DNA, what it do? It activate or it switch on some pathological genes which are responsible for the formation of the cyclooxygenase, cytokines, and phospholipases. So the phospholipases, cyclooxygenases, and cytokines are responsible for the inflammation and various pathology or pathological processes inside the cells or inside the body. So what the cortisol do here? The cortisol from this over here a response element. The cortisol combined with the response element along with the dimer, and this response element combined with these transcription factor, with such transcription factor, and inhibit these transcription factors. Inhibit these. For example, this is NF-KB, so this inhibit the NF-KB. So when the transcription factor are inhibited by this by this response element, when the transcription factor or pathological transcription factor are inhibited by the this response element, so this phenomena or this process, this is called switching off phenomena. Switching off phenomena. So the cortisol switch off the pathological genes actually, because this transcription factor will activate the pathological gene inside the DNA. And now when this transcription factor is inhibited, so this will no more activate the pathological gene. So we can say that the cortisol indirectly inhibit the pathological genes which are responsible for pathological processes inside the cell, which are responsible for the inflammation, for the analgesia, for the algesia, for the pyrexia, for the inflammation inside the cell. So the cortisol actually switch off all the pathological genes inside the cell. It work intranuclear, and it goes intranuclear, and it switch off all the pathological genes. It also switch on some physiological genes inside the cell. It's also switch on. It also combine with some transcription factor which are physiological. This response element also combined with some physiological transcription factor, which are responsible for normal physiology, and switch on the normal physiological genes. So the steroids are actually responsible. The cortisol, which is a prominent example of steroid, endogenous steroid, this is responsible for the reduction of inflammation, for the inhibition of inflammation, for the inhibition of pathological processes. Or we can just say that the cortisol is responsible for the switching off of all pathological processes, or switching off of the gene responsible for pathological processes, and it is responsible for the switching on of the normal physiological genes.